the protein by itself is partially protective in brucellosis. But brucellosis is a very complex disease where you need a strong humoral and strong cellular responses. So in the case of brucellosis, what we obtained was decorated with other antigens. We obtained a very highly protective response in ovine brucellosis, and we are doing the same now in bovine brucellosis. But we also demonstrated that using partially protective antigen in another model diseases, for example in rotavirus, when they are presented in this setting, they become more potent and more protective. So we have several models of protective effect in mice models, in experimental models. We are trying now to develop this in application models in veterinary and human medicine.
We have time for more questions. Just one curiosity, the Shiga experiments that you mentioned,
yes.
do you want to tell
yes.
us?
This was done in collaboration with Marina Palermo for Academia Nacional de Medicina, and in this case the challenge was to try to increase the immunogenicity of the beta subunit of the Shiga toxin. The Shiga toxin has an alpha subunit that has enzymatic activity and a beta subunit that has the capacity of attachment of the toxin to gangliosides in the membrane of cells. And this beta subunit is very weak immunogenic. So we were working for almost 2 years to try to express this pentameric subunit over our decameric structure, and we were successful. We think that we have 2 pentamers presented at each side of our decameric structure, and the good thing that this particle is highly immunogenic. We get 10 to 100 more antibody response compared to the beta subunit isolated, and these antibodies, when are used, are transferred to other mice, are completely protective. So this is a very interesting case where we took a weak antigen and we have converted it in a very strong antigen. We are now exploring the possibilities of using this protein particle expressing and presenting the subunit beta of the Shiga toxin as a vaccine, and also to use it for prepare antibodies of different type as a positive transfer in immunotherapeutic.
Okay. Any comment? Okay, let's go then to the next talk.